Welcome everyone. My name's Jan Strugnell and I'm the Director for the Centre of Sustainable Tropical Fisheries and Aquaculture, or the CSTFA. I'd like to welcome you to our seminar series. The CSTFA is a research provider for sustainable tropical fisheries and aquaculture research globally, and we provide world-class multidisciplinary solutions-focused research for Australian and international resource managers, both in government and the private sector. And we're based at James Cook University in Townsville, which is in far north Queensland, Australia. And I encourage you to check out our website to find out more about us, or also send me an email. Now today, it's my great pleasure to welcome Dr Alberto Rovellini to speak to us. Alberto is from the University of Wellington in New Zealand, and he's a marine ecologist interested in linking the ecological roles of marine organisms to broader ecological processes by developing and applying ecological models. And his PhD research focused on the ecology of sponges in Indo-Pacific reefs between Indonesia and the Great Barrier Reef. And today he'll speak to us about modelling regime shifts of coral reefs to sponge reefs. Thank you so much, Alberto, and I'll let you take it away.
Thank you so much, Jen. Hello, everybody. Thanks for joining us. Today, I'm going to give you an overview of my PhD research. So what I've been doing for the last 4 years here at Victoria University of Wellington, together with Professor James Bell and also Matt Dunn from NIWA and Beth Fulton down in Hobart with CSIRO. So I want to start introducing sponges a little bit. Marine sponges, so phylum Porifera, they are an exceptionally diverse phylum. So there are well over 9,000 described species, and they're also functionally very diverse. So most sponges actually host a lot of, like a pretty complex microbiome. Some sponges host photosynthetic microbes, so photosymbionts, in similar ways as corals do, in a sense. And there is a lot of morphological diversity as well. So you have a broad functional diversity. And regardless of whether sponges host symbionts or they don't, all sponges filter seawater. So what you see here is a video of a barrel sponge on a coral reef in Indonesia, and that's a fluorescent dye. It's harmless for the sponge, but it gives you a bit of an idea of the amount of water that these animals can filter on the reef. It's up to cubic meters per day. And as they do so, they take up bacterial plankton and picoplankton, so particulate organic carbon and also dissolved organic carbon. As they take up this carbon, then they effectively recycle it into the food web from the water column, because they are preyed upon by vertebrates, so fishes and turtles, and also by invertebrates, so nudibranchs and some echinoderms. In terms of interactions with other benthic organisms, they compete for substrate, and they do so quite effectively as well. So they compete with corals, they compete with macroalgae, and they compete with other invertebrates, including sponges themselves, so other sponges. And they give both positive and negative contribution to reef structure. So encrusting forms can stabilize the coral rubble by bonding it, and they can also provide microhabitat to organisms that live inside of them or around their skeleton, as you see for something as branching as this. I use the word skeleton loosely, more on that later. And some sponges contribute negatively to reef structure. So in this picture here, you see the yellow tissue is a bioeroding sponge that is boring into coral carbonate. So in the context of climate change and corals declining on the world's reefs, people have started looking how sponges do with warming and acidification. And some coral reef sponges seem to bounce back from bleaching quite easily, and they just switch to the heterotrophic feeding. And in fact, an increased PCO2 can enhance photosynthesis in some species. Plus, they don't have a carbonate skeleton, so they resist better than corals to acidification, or then they're not as affected. And a lower pH, in fact, facilitates bioerosion of coral skeletons. in saying that they are still sensitive to an increasing temperature and decreasing pH. So that works to an extent. So it's a complex response, a complex scenario. And it's been observed in the last few years that after acute events, for example, acute thermal stress in the Caribbean or in Brazil and in some locations in the Indo-Pacific as well, there's been an increase of sponges and a sort of a shift of coral reefs towards a more sponge-dominated state. And so people have started asking the question, is it possible that there is going to be in the future a shift towards sponge reefs? Usually when you think of a regime shift, you think of algal systems. Is it possible that you can get something like that for sponges as well? And what could the effects of a shift like this be on the ecosystem? And one thing that could help in answering these questions are ecosystem models. And one such ecosystem model, for example, is the Atlantis model. So Atlantis was developed by Beth Fulton, who is now a CSIRO. And it's an end-to-end model. End-to-end means that it represents all components of a system, from the biophysical components, so you have physical drivers, you have ecological processes, to also the human components. So you have industries, you have socioeconomic components, and so on and so on. And basically, you track the energy flows through the system, and you can use it to project scenarios. There are quite a lot of applications of Atlantis as of now. This map shows you the ones that are either already in place or far along in their development. There's more that are not shown on this map that are currently being developed as well. And the problem that you have when you think of modeling sponges in ecosystem model is that hasn't really been done that much so far for a couple of reasons. First, you have limited data a lot of the times when it comes to marine sponges, and limited knowledge as well of the ecological processes of sponges and their systems. There's also limited commercial interest in sponges. So a lot of the times what pushes the representation of a species in an ecosystem model is, for example, fishery values, and you don't really have that with marine sponges most of the times. And so they're often modeled pooled with other benthic filter feeders. But as I was shown before, they do have a lot of important ecological roles, and they are quite abundant, and they could become more abundant in the future potentially, at least comparatively to other benthic organisms that could decline instead. And so it would be a good thing to include sponges explicitly in ecosystem models, but there are still knowledge gaps before we can go ahead and do that effectively. And so in my thesis, I tried to address a few of these key gaps. One of them being, how do sponge assemblages develop over time? So an ecosystem model helps you project populations of organisms, for example, in the future. So you need to know how your population behaves over time, how it increases and how it declines before you can effectively model it and validate your model as well. What are the key processes, ecological processes of a sponge-dominated reef system? We don't really know that yet because there are not so many real-life examples as of yet. And finally, how may coral reefs respond to an increase in sponge dominance? So how would coral reef processes be affected by an increase in sponge dominance? So as for the first step, I'm focusing here for the most part on Indo-Pacific coral reefs, which are, and sponge assemblages, which are diverse, ecologically speaking, as I will get to in a second. So how could these sponge assemblages develop over time? There isn't a whole lot of information of how a complex sponge assemblage behaves in time. For the most part, long-term monitoring studies are carried out on corals, with a focus on hard corals. And so my objectives here were to quantify the temporal changes in sponge abundance and diversity, and to identify the relationships between these two variables over time. So the study location is southeast Sulawesi, which is in the middle of the coral triangle, in a place called Hog Island. So that was the home reef here, just west of Hog Island. And back in 2005, 3 sites were established on this reef. So this is what it looks like from a frontal view. You have 3 sites about 10 meters deep on a reef that is basically vertical, and the sponge assemblage over there is comprised mostly of small encrusting sponge species, which were counted in situ every year by the same observer for over a decade, using permanent quadrats, which allows you to basically track the same bits of reef down to the same individual sponge over time. And so basically I tracked the development of these 15 individual quadrats with mixed effects models to see what's been going on for the last decade. And the next thing that I was interested in is how, okay, not only the number of sponges, but also which sponges we get as the numbers fluctuate up and down. So the changes in species composition over time. And you basically have three potential scenarios, right? So this is your starting point. And if you have an increase in sponge abundance, you may have more of the same, basically more or less in the same proportions, the same species. You could have one or a few species that kind of take over and the other ones disappear, so the evenness of the community declines. And you could also have more species popping up and driving up your numbers, so the richness and the evenness of your community both increasing over time. And that's interesting because when you think of an increase in sponge abundance, you need to ask to yourself, like, which species are we gonna see? Is it gonna be a lot of the same ones, or is it gonna be a more diverse assemblage? And what we saw was that sponge abundance was fluctuating a lot over the study period, from minimums of about 50 sponges per meter square to up to 250 even. And it was the same pattern across the three sites, more or less the same pattern, although in different amounts, obviously. It was a very diverse assemblage, like I said, over 140 sponge taxa, which is quite exceptional, but in the Indo-Pacific, that's often what you find. And the species composition also changed over time. So it wasn't just sponge numbers. So in this plot, you see basically the composition of your assemblage. In the first half of the study period, it was up here, so to speak, 2005, 6, 7, 8. And then you have a shift when, just gonna go back, when you are in the lowest numbers, you have a shift in 2011 and 13. So you do have your sponge assemblage changing in its composition as well. And the more sponges you get, the more sponge species you get, true, but also the less even your sponge assemblage is. And that's an important point that I'm going to touch on again in a second. So to summarize the main take-home message from this, we do see a potential for a sponge assemblage under the right conditions to really increase or decline in terms of numbers. So there is a possibility for if free space does open on the reef as a result of, for example, a thermal event or whatever could happen, there is a potential for these sponge assemblages to develop. And 7 years is not a long time, and you have declines and increases for up to 60% in terms of sponge abundance. You do have a change in species composition, though, and what does that mean? It means that if your species evenness declines, it means that you have a few dominant taxa that really drive the sponge demographics at your site. And in this case, the dominant sponge taxa were mostly Protosuberrites, Cycons, so small-bodied encrusting species that can really boom and then collapse in terms of the numbers. So it's sort of like potentially an unstable or a really dynamic system anyway they're describing, and it does have a potential for a rapid temporal development. Which then brings me to the next point of focus of my PhD research, which was, okay, like, if you do observe an increase in sponge numbers, potentially in sponge biomass percentage cover and so on, what are the ecosystem level effects of this? What could this entail for the rest of your system, for the rest of your coral reef? To answer that question, you basically have to first figure out what the key ecological processes of your system are going to be, right? And like I was saying before, we don't quite know that yet, in that we don't have many instances of these sponge-dominated systems to observe yet. And so something that could help you is if you develop a conceptual model or a qualitative model, in this case of a coral reef. And I'm going to explain in a couple of slides what the qualitative model was and the capacity that I used it. And you could use that model to model or project basically the qualitative responses of your reef ecosystem to increases and declines in benthic, to changes in benthic dominance, so increases or declines in sponge abundance in our case. So when I say qualitative model, it is qualitative. It doesn't tell you by how much things change in your system, but it tells you the direction of your change. And qualitative model here is a mathematical framework which is based on matrix algebra, and it's cheap, like both in terms of it's not time consuming, it's not data intensive, and it's quick and easy to develop. It has a disadvantage that it only obviously yields a qualitative prediction, and it kind of is to an extent a reflection of your knowledge of the system as well. And the purpose of it is to formulate hypotheses and to identify gaps in your knowledge of the ecological processes that are at play. So if you imagine a simple system, basically you need to select the key components and the key interactions of your simple system. So if you imagine, a simple predator and prey system, you can represent this with a community matrix like this. And the community matrix basically details the effect of each component of the next, right? So the effect of the prey on the predator would be a positive one, and the other way around of the predator on the prey would be a negative one. And that's really what you focus on. It's not the magnitude that much of the effect, but it's the sign of the interaction, it's the sign of the effect. And in the next few slides, you'll see quite a few of these notations. So that's called the sign, the digraph. And it's basically the nodes, the circles or nodes are the components of your community, and the arrows are the interactions between them. So an arrowhead is basically a positive effect, and this circle here is a negative effect instead. And you'll see quite a few of these, and this is just a representation of a community matrix. So you can take that and you can scale that up in terms of complexity, and you can have something that looks like that, where you have a lot of nodes and a lot of interactions. And what you can do, you can impose a perturbation, which is defined as either an increase or a decline in one or a set of components of your system. And your goal is to see how this perturbation propagates throughout your system, given the shape of the interactions that connect your nodes, essentially. And you do that by performing what is called loop analysis. So you see like how the system qualitatively responds and how big your prediction uncertainty essentially is. So you'll see a few of these graphs from here on out. And when you see a blue node, it means that that component has a positive response to the perturbation. When you see an orange node, it's a negative response. Gray node, it's an uncertain response to the perturbation. So we sat down with colleagues of ours from Ames and from University of Auckland and from here at Victoria and CSIRO as well. And we realized a qualitative model, QM, for a coral reef, where we have basically a central benthic module with sponges, coral, and algae all competing for substrate, and corals contributing to habitat structural complexity up here. And then you have generic upper trophic levels that feed on these components. You have generic lower trophic levels that are fed upon by these components. You have detrital components in your system, and you have some key environmental drivers. So this is obviously a coarse simplification of the coral reef, but you only focus for this exercise on the key groups that you're interested in and the interactions between them. And you attempt to summarize the main flows of energy and matter between them. That's the notation that you will see from here on out. And the perturbation that we applied here was basically to simulate environmental degradation and climate change. So an increase in turbidity, an increase in acidity, and an increase in water temperature. So the way our model responded to when we tried to impose those combined perturbation was very uncertain. So we did see a decline, a negative response from corals and from habitat complexity. But from the rest, there was a very high mathematical indeterminacy. So how do you solve that? You simplify your model, and that's one of the qualities, one of the perks of qualitative modeling, in my opinion, is that you can basically break it down, which is what we did, into 4 models. One of them was basically a core model with only the benthos and the upper and the lower trophic levels. And then a model for the structural complexity, which is basically the same as the core model, but it also includes the habitat complexity here. A model that also includes the detritus loops that are connected to sponges. And one last model that always assumes that macroalgae have a competitive advantage on sponges. So basically, that always assumes that macroalgae smother sponges. And so if you simplify your system, you reduce the uncertainty substantially. And what we saw from the predictions of these 4 simplified models was that the perturbation expressed as an increase in sponge abundance in your system basically brings to an increase in sponge-mediated carbon flows. So you see an increase, for example, in sponge-eating fishes and in that branch of the food web, and a decline in corals, assuming that sponges mediate negative effects on corals by outcompeting them or by eroding them, some sponges. We also saw, and that's interesting, and I'll get back to that later, that the model that includes habitat complexity had quite ambiguous outcomes. So even if you increase sponges in this model, you don't quite know how the upper trophic levels are going to respond, because the decline in corals translates into a decline in your habitat complexity. And so what you would gain in terms of increased carbon flows from an increase in sponges kind of gets buffered by a decline in habitat complexity. So you have a broad uncertainty in your system. The model that includes the detritus pathways sees an enhancement basically in these pathways and in the carbon flows connected to these pathways. So again, an increase in spongivorous fishes and top predators, as in fish that feeds on spongivorous fishes as well, and an increase on the detritus in the system as well. And also interestingly, if you always assume, which is not always the case, but if you always assume that macroalgae do have a competitive advantage on sponges, then you also have an ambiguous response in many components of the system. And one of the main points that we will need to clarify moving forward will be what is most likely that we move towards a macroalgal dominated state or a sponge dominated state in terms of the regime shift. And that depends really on the contextual ecological situation of your reef. So as take-home message, a decline of corals and an increase in sponges projected by my qualitative model showed that you see an increase in the detrital pathways and an associated potential change in the reef food webs. You see kind of an ambiguous response of the system to decreasing habitat complexity, because you don't have that supporting the top trophic levels anymore, and in general the fish in your food web. And you do see also an uncertain outcome from interaction with sponges and macroalgae. So one of the points that I found most interesting from this exercise, so one of the gaps that the qualitative modeling highlighted was, can sponges kind of replace, to an extent at least, corals as providers of habitat complexity or not? And so with a field study, we went ahead and we basically measured with underwater 3D photogrammetry the structural properties of bits of reef that were dominated by sponges and bits of reef that are instead dominated by corals. And then we compared the properties of these reef areas. And so we identified whether sponges are good providers of habitat complexity, and in particular, which sponge growth forms or which sponge morphologies are good providers of habitat complexity. And we also did this in the Wakatobi. It's the same location, different reefs, slightly further up north, but it was still on Hog Island. And we went and laid down 32 large quadrats on the reef over there, targeting bits of the reef where either sponges or corals were the dominant benthic organism. And then within these quadrats, basically, we identified the most common coral growth forms and sponge growth forms. So you have, branching folios, tabular, massive, and so on for corals, and same for sponges. And we measured so we quantified the percentage cover of these morphologies and of all the other benthic groups in the quadrats. And then we built 3D computer models with a technique that was proposed by Young and colleagues in 2017. So it's quite handy and quite portable, and divers can do it in situ underwater with just an action camera and quite limited gear. It's quite time effective, and you can achieve good resolution 3D models. The example that you see here is a pretty large barrel sponge in one of the quadrats. It's one of the models that we took underwater. And then from these 3D models, we basically used Python scripts provided by Young and colleagues, and we quantified 7 metrics of habitat complexity. So very briefly, these were rugosity, which gives you an idea of like the reef contours and how complex they are. Vector dispersion, which gives you an idea of how heterogeneous the surfaces on your reef are. And then 5 metrics of fractal dimension, which basically each of those 5 gives you a measure of the size of the refuge holes on your reef, with D1 being the largest refuge holes, from 60 centimeters to 1.2 meters approximately, and D5 being the smallest category of refuge holes, so 1 to 5 centimeters, and then everything in between. And so basically the idea was to compare between coral-dominated and sponge-dominated quadrats at different levels of complexity, so at high and low complexity corals to sponge, and also within coral-dominated and sponge-dominated, so higher complexity corals and lower complexity corals, how complexity metric change and how habitat complexity changes across a gradient. And then also to identify which growth forms contribute to such habitat complexity, with variable selection tools. And so approximately of those 32 quadrats, we had 50-50 between coral and sponge dominated. So we did a good job in picking reef areas that were either sponge dominated or coral dominated. Percentages were quite high for both of them. So the way we organized the high and low complexity wasn't arbitrary, but we did a clustering routine based basically on the 7 complexity metrics. So again, the idea was that at this stage, we were able to compare high complexity coral quadrats with high complexity sponge quadrats, where high complexity is based on the 7 metrics, and same for low complexity. So there's a lot in this picture, but basically each of these 7 panels is one of those 7 metrics that I was talking about. And if you just focus on the right-hand side of each of these panels, that's where you have high complexity quadrats. And what we saw was that, interestingly, at high complexity, there was no difference in rugosity between sponge and coral quadrats here. This is flipped, like the lower this metric, the more rugose actually your quadrat is, but so it's not decreasing, it's actually increasing. Same goes for vector dispersion. So there was no difference between coral and sponge quadrat. What got interesting was that if you look at the size of the refuge holes, it was small refuge holes were more abundant, large refuge holes were more abundant in sponge dominated quadrats, and small refuge holes were more abundant in coral dominated quadrats. In terms of which morphologies explained the overall 3D structure of my quadrats, permutation tests showed that basically we had branching and massive coral colonies that explained basically the overall 3D structure of the coral-dominated quadrats, and mostly barrel sponges instead in sponge-dominated quadrats. And then when we tease apart which metrics are, which morphologies contribute to which metrics, we saw that massive and branching corals contributed mostly to rugosity and vector dispersion, but most importantly was the branching colonies that contribute to those small scale refugia for coral-dominated quadrats. And instead for sponges, we had barrel sponges contributing to those large refugia instead. So take-home messages from this was that, yes, overall, we can compare sponge-dominated and coral-dominated bits of the reef in terms of the overall 3D structure, but sponge reefs in this study offered more large refugia. When I say large, obviously, we're talking mesoscale, because these were 4-meter square quadrats. So it's not, by no means, it's large-scale habitat complexity. But really, the barriers were what contributing to these habitat complexity in the sponge-dominated ones. And then without the branching coral colonies, you don't really have those small-scale refugia on your reef. And whatever habitat complexity you have provided by the sponge, it doesn't really survive the sponge, because sponges don't leave behind a skeleton like corals do. Sponges degrade very quickly when they die, as you see in this picture. And within a few days or weeks, there is nothing left, no matter how complex the sponge was in the first place when it was alive. So yeah, in the long run, it's unlikely that sponges can replace corals as providers of habitat complexity. So all of this brings me to how we included this information in the Atlantis ecosystem model. So as I was saying at the start of this webinar, sponges do have a number of important ecological roles on coral reefs, and they may, at some locations at least, become more dominant in the future. But they're often quite oversimplified in ecosystem models like Atlantis and Ecopath. And if we build a model to project coral reefs in the future, it's important that sponges are modeled more explicitly than they've been in the past. And so basically we incorporated the information that I've showed to you so far into Atlantis. So as a short reminder, Atlantis is a complex model, and it includes a lot of moving parts, from the biophysical to the human components that are closely interlinked with one another, from the biophysical, the physical, and the oceanographic, to the socioeconomic, and even the assessment components of your ecosystem. So there is a lot going on, and we wanted to capture what we know about sponges a little bit more explicitly than it had been done in the past. So the first step was including, like everything that is known from the literature. So the functional diversity of sponges as a group of coral reef animals, in general marine animals, because there are also temperate Atlantis models. So phototrophic sponges, bioeroding sponges, the dock cycling, and all those functional roles that sponges have. We wanted to include the dynamic aspect of sponges in Atlantis. Atlantis projects population dynamics into the future or into the past if you're doing hindcasts. And so it's important that you can validate those dynamics that the model produces against something. We wanted to include all the interactions that sponges have with other benthic organisms, namely macroalgae and corals. Well, most interestingly, in my case at least, with those groups, but also with other benthic groups, and also the interaction that, top-down controls from predators on sponges. And then we wanted to capture this contribution that sponges can give, differently from corals, but still a contribution to habitat complexity on coral reefs. And so the way we included all this information in Atlantis, we allowed for 3 sponge types, so to speak, in Atlantis. We built on the coral code, and I did this work together with Beth Fulton down in Hobart. So a lot of infrastructure was already there in the model, and we allowed for the representation of heterotrophic, phototrophic, and bioeroding sponges to cover a little bit of that functional diversity that sponges have. I'm not going to go into details of the Atlantis code because it's quite abstract, but the main highlights are that we basically were able to implement a three-way competition between sponges, corals, and macroalgae, where for each species, basically, that you model in your Atlantis application, you can decide the competitive outcomes of these interactions. So you can decide whether algae smother sponges, whether sponges outcompete corals, and like you can fine tune all these interactions. And on top of that, you also have an underlying competition for substrate with the other benthic groups that are represented in Atlantis, which include, soft corals and other filter feeders, turfine algae and CCA and so on and so forth. We also included a submodel to allow for sponges to contribute to habitat complexity. So Atlantis does capture habitat complexity, and on that depend, for example, fish recruitment and the way some fish groups may escape predation as well. And these are all things that you can completely parameterize to your needs in your Atlantis application. But we wanted to at least have the option to allow for sponges to contribute to overall reef architecture, and also for sponges to contribute negatively to reef architecture. So basically by eroding. So sponge by erosion wasn't explicitly covered in Atlantis previously, and so we wanted to include that because it's been shown that in areas like the GBR, the Great Barrier Reef, for example, at some locations at least, by-eroding sponges may, to an extent, increase in abundance. And more functions, silicon limitations, nutrient limitations, bottom-up limitations that are very important, and I'll get to that in a second as well. And I tested these new sponge functions, this new sponge code to by contributing to the development of an Atlantis application for the Great Barrier Reef. All good, sorry. Right, so just a couple of words about Atlantis Great Barrier Reef. This is a work in progress still, and it was developed at CSIRO by Beth Fulton and her team. It's a large model, so it has over 100 spatial polygons, and that's how Atlantis models are organized. So these cells that you see here are basically sub-areas of your model geometry. And it has almost 90 functional groups as well. And the calibration exercise is still ongoing for this model. And there was a lot of data that went and is going into this model. I mostly worked on the benthic component, and we mostly used the data from the AIMS long-term monitoring, for example, and other monitoring programs that have been done in the past on the Great Barrier Reef area. And then there is a whole lot of other data that have gone into the model for model geometry, for example, for the hydrodynamic models, biogeochemical models, fish base for fish stocks and stock assessments and all that. I'm not going to go into details, but just to give you a rough idea of how large an Atlantis application can be. The way I included sponges in this model was three groups, so heterotrophic, phototrophic, and bio-eroding sponges. So three main, if you will, functional groups of sponges on the Great Barrier Reef that have been characterized in the literature. And then we also included one group of specialized spongivorous fish. And mind that all groups, depending on how you parameterize your Atlantis models, can, to an extent at least, feed on your sponge groups, for example, occasional feeders and opportunistic feeding and all that from invertebrates. So calibration and validation are ongoing, like I said, but the starting point of your model looks like that. So basically you interpolate the data that you have to the model geometry that's proportional cover, for example, in the Atlantis boxes for your three sponge types. And they have different distribution depending on the data that you have and depending on the literature that you use to build your model. Okay, now very quickly to wrap up, I would like to spend a couple of words on where this all leaves us. So what is the possible ecological functioning of sponge reefs? So we've seen that sponge reefs may be a highly dynamic system. So there is a lot of temporal variability of sponge populations. You may have an increase in sponge-mediated carbon flows if you have an increase in sponge abundance, and you may have a loss of small-scale structural complexity if you have an increase in sponge abundance and a decrease in coral abundance and coral cover. So you need to, it is good to look at the species composition of these systems that change over time, because when you have an increase in sponge abundance, it's important to see if you still have high species diversity going on, or if you have just a few species that are taking over, which was what was happening for the most part in the case study that I presented here. And so that brings the questions like, if you have species-specific associations, for example, of those species, how are those going to flow on into the system, right? If you have an increase of some of the species and not of others, and how are the other organisms that are connected to those species going to behave and going to respond to these changes in abundance? When you have an increase in sponge-mediated carbon flows, you may have an increase in sponge-produced detritus, you may have an increase in sponge-eating animals, but you need to keep in mind that there is food limitations for sponges. So are the nutrients going to be able to sustain sponge populations if sponge populations increase or not? And how is all of this going to interplay with the loss of small-scale habitat complexity in your system? So you basically also need upright sponge morphologies to keep this habitat complexity. If you have mostly just encrusting forms, you won't retain even that complexity that sponges can provide, at least in terms of larger refuge holes. And sponges can be damaged by storms and cyclones. So if you think in a context of environmental change, these are predicted to increase as well. So on shallow reefs, you might actually not have those 3D sponge growth forms that sustain your complexity. And if you do lose those small refugia, you know, like in my case, it was between 1 and 5 centimeters. So everything that is smaller than 5 centimeters, which is a large chunk of fish biodiversity on some reefs, may be impacted as well, and that may have flow-on effects as well on the system. So ultimately, the function of the ecological function of these reefs, of these assemblages, will depend largely on which species will become dominant. And regarding that, there are substantial caveats. So sponges are still sensitive to warming and acidification. So there may be a window for sponges to sort of like resist these scenarios more than corals, but there will be a threshold after which you will have effects of warming and effects of acidification on sponges. There will be substantial bottom-up limitations. So if you think of stratification of the water layers on shallow reefs, you may not have that income of nutrients, for example, from deeper waters that may sustain your sponge populations. You may have top-down limitations, obviously, from things eating sponge populations that are increasing. You may have competitive limitation to sponge populations increasing in the first place. So you may have the system shifting towards an algal-dominated state instead, as it's been largely observed at several locations for the past few decades, essentially. And we also need further research to basically refine how we model sponges. So we need to better define the ubiquity of sponge-associated detrital pathways. So whether detritus, whether dissolved organic carbon basically is recycled through the sponge or not, and where that happens, on which reefs that happens. We need more information on the physiological responses of sponges to climate change and on the competitive interactions with macroalgae. Not a big deal of work has been done yet, especially in the Indo-Pacific, on how these two groups interact with one another. And same goes, like I was saying, for the bottom-up limitations to sponge growth. So yeah, that is the end of my talk. Thank you very much for listening to me, and I think I'll take questions now.
Great. Thanks so much, Alberto. It was really interesting. If you have any questions, please write them in the Q&A. So that's down at the bottom of your screen there, and we'll get them to Alberto that way. I just have one to start off with, Alberto. I guess I was wondering, obviously sponges don't fossilize as well as corals, but I was wondering what evidence there might be from the fossil record of shifts from coral to sponge reefs or vice versa. So just historically.
Yeah, so that is one of the things, one of the indications for the fact that there may potentially be a shift towards higher sponge dominance is that in the past there have been the oceans have been a whole lot more acidic and warmer than they are in present days, and it has been argued that from fossil records, from spicules essentially, it wasn't corals, it wasn't in general carbon building organisms that were dominating the reefs back then, but it was more silicon based organisms like sponges. Obviously it's not the same thing because changes are happening very quickly now, and you're talking about changes that had been happening over geological times back then, but you know, on a general theoretical level, because it's been observed that in those more acidic and warmer, basically, oceans, there was a higher sponge abundance. One could well think, like, if this is going to happen again in the future, are we going to witness a progressive shift, and or at least a disproportionate negative impact of warming and acidification on sponges compared to the impact that it has on corals?
Okay, thank you. So just another question I was wondering, maybe you touched on this and maybe it's not that important, but to someone who doesn't know much about sponges like myself, it just seems that they'd be super hard to identify to species. And I just wondered the ability to, you know, is that a problem with this kind of work, or is sponge taxonomy easier than it looks like it might be?
No, it's yeah. Case in point, it's difficult. It's difficult for a number of reasons. It's a relatively young branch of taxonomy, and I'm not a taxonomist by any stretch of the imagination. You do need a molecular means to be sure of your sponge identifications. So it's kind of a more expensive process and a more time-consuming process than IDing other taxa, and you can identify sponges to an extent from spicules, but there will be a quite coarse identification, and the ideal case would be merging morphological identification of spicules, for example, with molecular methods, because a lot of the times you have species that look alike a lot, but are not the same species at all. So you have cryptic species within the same you know, morphotypes, and on the other hand, you have maybe in different geographic location, you can have a quite marked dimorphism within the same species. So you might have the same sponge that looks substantially different in different places, and you might categorize it as different sponges, whereas where it's actually the same. So yeah, definitely it's not easy, and that is one of the reasons why it hasn't been easy until now to include that information about sponges in ecosystem models as well. When you are focused on one or a few species, it might be complicated to find reliable data on a large geographic scale, at least on those species.
Thank you. We have a question here from Nicholas Cornder, and he asks, in your eyes, which gaps of knowledge or data drive uncertainty in coral reef models that attempt to incorporate sponges more explicitly?
Yeah, so I think it kind of nicely ties back to what we were talking about. So sponge taxonomy is complicated, and so I guess that in general, sponge science is lagging behind compared to coral science a little bit. Things are changing now. There is a lot of interesting sponges at this time, but we just don't have the ground knowledge for a lot of sponge related processes to be able to actually tie them into ecosystem models meaningfully. And that was the reason why I did a lot of the work that I did. So the qualitative model that we developed, for example, in one of the lines of work that I just presented, the purpose of that was exactly that, to identify the main gaps, the main things that we don't know yet as far as sponge ecology goes. For example, there is the so-called sponge loop, which in 2 words is basically how sponges take up dissolved organic carbon and turn it into detritus. And by that, they basically put it back into the food web, because that detritus gets eaten by detritivores. Now, that hasn't been, the ubiquity of this process hasn't been demonstrated yet. So a lot of that work has been done, for example, in the Atlantic Ocean, in the Caribbean, and in the North Atlantic as well. There isn't a whole lot of that on the Great Barrier Reef, for example. So it's unclear how sponges in different locations cycle dissolved organic carbon. And because that is such a big chunk of the food web, potentially, if you then try and model sponges as part of a food web with an ecosystem model, you have a big question mark as to what happens with detritus and with docks. So that will be one of the main gaps, definitely, that need to be addressed in the near future to make sponge models even better, including the way we include sponges in Atlantis, for example.
Are there any other remaining questions for Alberto? Well, I just let people have a think about it. I'll ask just maybe a really small one. I guess I was wondering also if we know how long most sponges can live, is it possible to age sponges?
Yeah, it's difficult to age sponges. Mixed bag. So what you saw in when I was showing those dynamics over time, for example, those would be short-lived sponges, and a lot of species would even be seasonal, actually. So you would have summer booms in some temperate systems, for example. And on the other hand, larger sponges, like barrel sponges, like the one that I was showing in the first video, for example, could live, depends who you ask, actually, up to decades for sure, perhaps even up to centuries. So it really is a mixed bag. They are, in general, quite slow growing animals, a lot of them at least. But they're not very easy to age at all. So you kind of have to, the way people have done it in the past, for example, even people here in our lab, was by doing photogrammetry, like 3D models on them each year. So if you can basically measure the increase in sponge volume every year, and that gives you an idea of the growth rates of the sponge. If you have cultures in a lab, you can obviously measure it a lot more easily, but in the field, it's not very easy. And case in point, that's one of the other reasons why sponges are a bit finicky to include in ecosystem models, because it's when you start thinking of how do you parameterize your growth rates and your demographic rates in general, you might run into problems, and you might have to do a little bit of generalization and a little bit of guesswork.
Off the hook. But thank you so much for your time, Alberto, and for giving us a great presentation. We really appreciate it. I know it's pretty weird talking into the camera for these Zoom webinars, so thank you so much.